So let's talk about the movement of light through the eye and then to the brain where it will be processed. One thing I just want to point out quickly is the eye is filled with fluid, right? We talked about how it is filled with an aqueous humor, it is filled with a vitreous humor. So if we've got our eyeball here, we know that it is filled with these fluid types, very thin in the front and very thick in the back. So as light enters the eye, in the same way we see refraction, like with this straw, we see that our vision is displaced a little bit. And what you're looking at are the waves of light being altered as the waves of light shift from traveling through the air to the water. So because the eye is filled with fluid, it's going to bend at certain points. The lens changing shape is a really important determinant of how that light gets through the eye and to where we have the highest concentration of photoreceptors, which is on the fovea. So when we're thinking about light entering the eye, again, all of the structures in the eye are there to focus the light on this fovea region where we have this high concentration of photoreceptors we refer to as rods and cones. And they're really just specialized neurons with different shapes to them. Rods fire signals to let you know when it's dark. So when your eyes are closed, your rods are on, sending action potentials. And cones are what turn on when you move into bright light. And those are the ones that allow you to see color. So we have a really high concentration of them back here on the fovea. We still have them in other areas which allow us to see peripherally. However, if everything anatomically is in its correct position, the light will enter up front here through this cornea area, through the sclera, and it will hit the lens, which, depending on the shape and size of the lens, will bend the light so it refracts at that when it enters the lens and when it exits. And then it's further refracted through this vitreous humor. And again, if everything anatomically is the size and shape and distance that it's supposed to be, we get the perfect focusing of light rays onto this fovea to give us clear vision. And we refer to then an eye that is anatomically sound, that has all the proportions to direct light to the fovea, as an emmetropic eye. However, there are also abnormalities that can occur in relation to the eyeball. When the eyeball is too long, so in this picture we see that we have what's called a myopic eye. And when you call somebody myopic, it means that they can only pay attention to their point of view. They're not seeing the grander picture. They're just paying attention to what's in the immediate. And that's what myopia is. You can see clearly up close. You're nearsighted. But that's it. So there has to be some sort of correction, because in myopia, where you can see, up close but not far away, the eyeball is anatomically, it's oblong. So it's not a circle that would cause the bending of the rays to the fovea. Here, where the fovea should be, we still have vitreous humor because of the oblong shape of this eyeball. So by the time these light rays become scattered at the actual location of this fovea, they're now going to be so far away that we have blurry vision. So this is what you would see in an uncorrected lens. That's why if this lens isn't in the right place and the right size and shape relative to the shape of the eyeball, then we have to add a corrective lens in front. And that's what you see here on the bottom in myopia. A concave lens, it scatters light, so we have this bending earlier. And when it's bent earlier, it will focus earlier, so that we have the perfect concentration of light rays on the fovea. So myopia, nearsightedness, is an anatomical deviation to the length of the eyeball. It's more football shape. And then hyperopia is farsightedness. So that is an anatomical variation where the eyeball is too short. So again, the lens is a certain anatomy, the cornea and the conjunctiva is a certain anatomy. So it is all set up for the fovea to be in the location where it is expected to be if the eyeball was a perfect circle. If the eyeball is too short, by the time we get to the anatomical fovea, we still see, like in the prior slide, that the rays are scattered. That will lead to blurry vision. And you see this more with, farsighted? That means you can see far away, so seeing up close is a little bit more difficult. So in this case, we use a convex lens, which is going out on both sides, and that moves the focal point forward to accommodate the eyeball being too short anatomically. So we have hyperopia, eyeball too short, and myopia, where the eyeball is too long. If you have both of those at the same time, that's called astigmatism. I'm lucky enough to have that, which means I get to pay extra for my glasses. Yay, maybe you do too. Congratulations. All right, so when I'm talking about the lens is this structure right here in the middle of the eyeball. This is not the iris. This is the ligaments that suspend the lens. So this would be in the center of the eye. And these muscles here, these are the ciliary muscles. And one important thing to remember about the ciliary muscles is that they contract in a circle. So just like a sphincter, I love saying that word, sphincter, they contract in a circular pattern. I want you to imagine that the lens, again, just like a trampoline, if this is our lens in the center, these ligaments that hold the lens in place are going to change in tension. When there is a lot of tension, so at rest, when nothing's going on, the lens, just like a trampoline, is going to be held suspended and flat. So you could go jump on it and bounce on it. That's a person bouncing on a trampoline. Let's make it flat now. However, if there is any slackening that occurs, so if we loosen up these areas, these springs that hold a trampoline, we're going to see instead that the trampoline bows down, and it's not going to be as tight and bouncy to go up and down. It's going to be thicker and fatter and a little bit more lax. So the ciliary muscles, by their contraction, are going to alter the tightness of these ligaments on the lens. So I want you to generally imagine that at rest, we have this trampoline-like configuration with taut ligaments and then a flat lens. However, when we have contraction, that loosens up the hold these ligaments have on the lens, which allows the lens to relax. And when it does, it gets really fat. It gets fat. And why that's important is in order for you to see up close, you need a fat lens. With aging, because the integrity of the tissues change, things tend to thicken and become more rough. So we have a change to these ligaments, and after a certain point in time, we're not able to get those variations to make the lens nice and fat, which is a correction that you need to do by adding reading glasses. We call that ability of the eyes to respond to close or far away vision. We call that accommodation. So accommodation, again, it refers to the ability of these ciliary muscles to contract in a circular way, and when they contract, loosen up on these ligaments and make this lens nice and fat. At rest, though, these suspensory ligaments, they're taut, they're tight, and the lens is skinny. All right, so in order to see up close, you need a fat lens. The muscles, again, they contract as a sphincter, contracted muscles, loose fibers, like our example of the trampoline. If we contract these muscles in a circle, it's going to loosen these up and allow the lens to relax. Whereas at rest, the force is pulling this taut. So relaxed muscles, you get a taut, tight, thin lens, but contracted muscles give you a fat lens, which you need to see up close. As I mentioned, when you get older, these suspensory ligaments start to harden and undergo fibrous changes, which make it really hard for this to change shape. So you don't get a big fat lens. If you don't have a big fat lens, you have to add one. So you add a reading glasses like this over here. That's our external lens. So that's added some more bulk, some weight to the lens. If it's not fat enough on its own, you have to add something to it. So remember, fat lens, seeing up close for just a second. And this is what you see with accommodation, with far versus near. Again, in looking at things either distant or far away, in order to see close, we need slack ligaments and we get a nice fat lens. So seeing up close, fat lens. If you are resting the eyes, if they are at rest, then you can see far away, because of the way that light refracts with distant vision. It focuses light on the fovea. However, if we have any abnormality in the ability of the lens to get fat and change shape, then we are going to have scattered rays on the back of the eye. So we need to add a corrective lens out here to add that bulk to the lens. When this happens to people around the age of 55 is the average age in the United States population when you average men and women, where you see a global experience of what we call paralysis of accommodation. Paralysis of accommodation is when you lose this ability due to these changes to the fibrous ligaments. So at that point in time, you would have to add reading glasses to see up close more adequately. All right, of course, there is some sort of autonomic input to this. And you might know, if you're familiar with the nervous system, that in sympathetic outflow, we have an increase in the size of the pupils. The pupils, they dilate. They allow more light to enter so that you can take in more information about a threat or anything you need to respond to quickly. So when sympathetic nervous system is activated, what it does is relax the ciliary muscles. And when they're relaxed, these ligaments are nice and tight. And when they're tight, that means the lens is relatively flat. So we have a flat lens, which means it's easier to see far away rather than right in front of our face, because usually the threat is, somewhere a couple feet from us and not sitting in our lap and staring at us like a book might be. So we get the relaxation of these ciliary muscles, which pull the lens nice and taut. There was one with the letter R. So here's our inverted image. So an R, when you're looking at it, looks like this. So we flip it and then we rotate it. So that conversely, in parasympathetic outflow, when there's too much parasympathetic activity, you get the opposite of dilated pupils. You get pinpoint pupils, which is called miosis. And this is when, again, the pupil is going to get really small. And in order for the pupil to get small, we have to have contraction of that ciliary muscle. And remember, when it contracts, it puts some slack on those ligaments, and that slack allows the lens to get fat. And the fatter the lens is, the better we can accommodate light entering the eye from things that are close to us. So in parasympathetic, we are if all's going well and the system's working perfectly and we have acetylcholine released and working its way up around the eyes, we have a baseline level of tear activity and we have the ability to see up close because of the contraction possible at the level of the ciliary muscle. All right, so parasympathetic, we get meiosis and we get muscle contraction, whereas sympathetic, muscle relaxation and dilated pupils, which we call mydriasis, if you're interested. All right, finally, when we are talking about light entering, so we already went through light entering and bending at these various places. The lens is going to change its shape. And it also, remember, these ciliary muscles, when they contract, they're going to contract in a sphincter-like fashion, which will also go along with changing the size of the pupil. So we went through that. So as light is entering, hopefully it hits the fovea, which contains those photoreceptors to tell us what we see in color, in black and white. And when that information is collected, it needs to go to the brain. There are some nerves involved in this. When the photoreceptors and ganglion cells and bipolar cells all converge together, they create a nervous tract, which is a cranial nerve called the optic nerve. It's cranial nerve 2. And remember, everything in the body is bilateral, so we have 2 optic nerves, 2 cranial nerve 2s. The way that these nerve fibers leave the eye and travel to the brain, first of all, is that on the medial aspect of each eye, so this is the left eye, I'm going to draw it in blue to go along with this color. The medial aspect of each eye, information from the medial or middle aspect is going to travel along and stay for a little bit on the left side, but then it is going to do what we call decussate. And decussate is a fancy way of saying cross. It is going to decussate or cross over to the other side of the brain. So this is the right, this is the left. Medial aspect, that nerve crosses over, it decussates, where it hits first the thalamus. So I'll get back to that in a second. The lateral aspect of each eyeball, in this case the left eyeball, does not cross. When something crosses, we say it moves contra or against, contralateral, against that side. So it moves contralateral. It doesn't stay on that side. It moves contralaterally. So the lateral aspect of the eye, these nerves converging from the rods and cones, the bipolar cells and ganglion cells, these ones stay on the same side. We call that movement ipsilateral. They remain on that side. They don't cross off. So the lateral nerves, they don't cross at the optic chiasm. Instead, they just travel along with their bros from the medial aspect, and then they part ways at the crossing at the optic chiasm. And the fibers from the lateral tract stay on that side. They stay ipsilateral. You'll see the same thing. Here's the right eye. So we have the medial aspect, these nerves coming from the rods, cones, bipolar ganglion cells converging together to make a nerve tract. They form the optic nerves, and the fibers from the medial aspect, the middle aspect of the eyeball, move contralaterally. They cross at this area called the optic chiasm. Whereas the nerves from the lateral aspect, the side of the eyeball, remain ipsilateral. They remain on that side of the brain. and they part ways with their bros, the other optic nerve, which is a cranial nerve, but they don't cross at the chiasm. They stay ipsilateral, and they travel on their own with the medial aspect of the other side of the brain until they get to the thalamus. And the thalamus is important with regards to all sensory processing because every sense but smell, touch, sight, hearing, all of this stuff, it undergoes what we call thalamic relay. The thalamus is the part of the brain that receives sensory information and categorizes it. It makes sense of it. I like to think of it as one of the old phone operators that when you would call, they would say, hang on a second, let me connect you. And they would pull a wire out of this large phone board and plug it into whatever the wire was going to, the outlet to who you wanted to talk to. So they're plugging it in to where, you want to go. That's what the thalamus does. So the thalamus organizes sensory information so that it can go to the part of the brain that's going to make decisions on it further with some more information in line. And why that's important is we can't make any other judgment about that sense because it goes to the thalamus first. It undergoes that thalamic relay. Because smell doesn't do that first, it goes to some other areas at the same time called the limbic areas, which are involved in emotion. You can have an instance of smelling something and it call up at a memory for you. Like maybe you remember cookies baking or something. Like when you smell those cinnamon brooms, you're like, oh, Christmas, I love the holidays. Because it doesn't go to the thalamus first. You have this mixing, essentially, of the sensory input with the emotional component that goes along with those other areas, which is important for other animals who use their sense of smell for things like predator stress. But for humans, like, we don't smell somebody we hate and be like, oh man, Bob's over there. I hate that guy. Like, we can't do that. I wish we could. But for us, it's what we call an evolutionary remnant, where we still have co-processing, essentially, of smell with emotion. So anyway, sight's not like that. You don't see a chair and think about every other chair you've ever, sat on, or recall your favorite chair at the same time. You just know it's a chair. That's because it goes to the thalamus first. And the area that it goes to in the thalamus is called the lateral geniculate nucleus. Lateral means to the side. Geniculate is the name of those cells because it's around a gyre that is named in that fashion. And then a nucleus is a group of cell bodies that all do the same thing. So once it makes its first stop at the LGN of the thalamus, these thicker nerves, so it now comes out as many nerve tracts, and those big tracts are called the optic radiations. And the optic radiations carry that processed information from the thalamus to these different nuclei in the more specialized cortical areas, and the specialized visual areas are in the very back of the brain in a lobe called the occipital lobe. And in those areas are things like the primary visual cortex, the association visual cortex, all the areas that help you put stuff together so that you know what you're seeing, you know what you're looking at. One other thing is, I don't think it's really on this picture, is it? Kind of. So when we have this thalamic relay, there's a couple of other nuclei right in this area. And it's a very base level instinctual type of place. And it's called the superior colliculi. We also have inferior colliculi that have to do with sound. But superior colliculi are pieces of the brain essentially that receive visual information that's not going to go over here so you can understand what you're looking at. Instead, this area, these superior colliculi, which are really close to the thalamus, on like a rodent brain, they pop off the top of them. They look like little dots. When that visual information goes to these colliculi, it allows you to have a reflex relative to something in your visual stream. What's the word for it? It poses some sort of threat to you. In the way that, like, if I threw a baseball, before you had the chance to process, like, Dr. B is throwing a baseball at my face. I don't like that. If the baseball hits my face, it's going to hurt really bad. You just duck, right? You duck from it. Or anytime something's flying at you or coming towards you, you have this really quick reflex reaction before there's any processing at the cortical level. So those things in the visual system are processed in these areas called the superior colliculi, and they are just meant to keep you alive. So they will orient you away and initiate reflexes to survive something flying at you, at your eye, at your face. All right, so those are the visual pathways. Light in the eyeballs to the fovea, through the rods, the cones, the bipolar cells, and then the ganglion cells converge together to form the optic nerves. The medial aspect of the optic nerves, they travel till they get to the optic chiasm, where they cross and travel then to the thalamus, whereas the ganglion cells forming the optic nerve from the lateral side of the eyeballs remain ipsilateral, traveling through the brain on the same side, but still are going to make their first stop at the lateral geniculate nucleus of the thalamus before traveling on through the optic radiations back to the primary visual cortex of the cerebral cortex. The only thing that's going to pop off of that are just necessary visual information traveling to the superior colliculi to initiate reflexes so that you don't get smacked in the face. The next one, stuff. All right, finally, these are just showing you some other pictures. Oh, look, you do have this stuff on here. So I mentioned our occipital lobe. That's where the primary visual cortex is, which helps you make sense out of what you just saw. We also see our first stop in the thalamus here, the lateral geniculate nucleus. Here, that is labeled, that I didn't know if it was there, and I'm happy that it is the superior colliculi. See, I drew them as little balls. That's what they look like, superior colliculi. All right, we also see the nerves colored appropriately and crossing over from the medial aspect at the chiasm level and staying ipsilateral from the lateral level. And when you're looking at the visual field, so here everything that is blue, so all of these things from the medial aspect going back to the, in this case, this is right, and this is left. So going back to the left side of the brain, those things enter from the right visual field where, so that's right eye only. So they enter the right visual field, whereas here's our left eye. We have the lateral aspect, medial aspect here. Everything in yellow is coming from that visual field. So the important thing is that what to take away from this, when you are looking at something, your eyes in this journey, they flip it. They turn it upside down and they make it backwards. And you can see that on a variety of slides. Let's go. Visual fields are showing you all this light is coming in from all different areas and it's focused on the fovea. And now your brain has got to make some sense of that. So it trades its information. So again, blue, it's going to come in here, it's going to travel down, and then we've got some left eye over here. So our visual field is going to cross over. And that's what you see. That's what the optic chiasm is for. It's a crossing point. that allows us to make everything make sense so that we see it in its correct orientation by the time it is processed by our primary cortex. It kind of rotates it all back from its sensory input so that we can make sense of it. All right, and this is what these things look like in a brain. Here's our optic nerve. I love the optic chiasm. It's such a great, clear piece of neuroanatomy. See this beautiful X there? Like, how beautiful is that? That's our optic chiasm. Here is the superior colliculi in humans. It's two little dots right there. Here's our contralateral fibers, our optic radiations. Beautiful, beautiful, beautiful neuroanatomy. Corber is this thing that separates right and left hemisphere. Callosum, you might remember. All right, finally, another thing about the optic chiasm is my favorite brain areas above it. It's called the suprachiasmatic nucleus. Supra meaning above, and chiasmatic meaning the optic chiasm. So this brain part exists above the optic chiasm. This suprachiasmatic nucleus, its job is to respond to changes in light. It's a great pathway. So we have a pigment called melanopsin, and it's in the same family as serotonin. So we often call it by its biochemical name, melatonin. And melatonin is made by a gland called your pineal gland, which is not in this picture, but if it was, it'd be way back here. So the suprachiasmatic nucleus, because it's right above that crossing of the visual fibers, its job is to sense if it's light out or not. When it is light out, there is an inhibitory signal to the pineal gland not to make melatonin. So as long as it's light out, you don't make any. But as soon as it starts to get dark, this is a moon, there's some stars. They come out at nighttime, which is when it's dark. When that happens, the suprachiasmatic nucleus no longer has that inhibitory signal to the pineal gland, which means it is dark. We don't have any inhibition, so the pineal gland is going to make melatonin. And when it makes melatonin, that makes you sleepy. Melatonin equals sleepy. So take into account that it's this part's job to sense light. That's why you're not supposed to look at screens like your phone or the TV, or even turn the bathroom light on when you go to pee in the middle of the night. Any kind of bright light signals to this very basic physiologic part of the brain, whose job is to sense light and then halt the production of melatonin. So we want total darkness. We want to not look at screens. Everything is perfect. And obviously, if it's not, you can supplement melatonin. When you take supplemented melatonin, you're essentially not making it, you're supplementing it for yourself. So it'll bind to the same receptors that melatonin or melanopsin does. And that daily rhythm is called circadian, which means around or about a day, which is a fun term because we know that the days are 24 hours, but when you put people in a room and allow them to rotate on their own circadian rhythm, they kind of settle around 23 and a half hours. And with our moon and sun, ours is about 24. So we're never quite right. And that might explain all of your life problems. All right, everybody, stay smart.